and Keshav for inviting me here. Over the next 20 minutes or so, I'm going to describe one of the activities that we have in the lab, looking at pharmacogenomic variations on the scale of population genomes and how we could translate it back into individualized therapies for individuals out there in the clinics. Before I start my talk, of course, some mandatory disclosures in terms of licensing and grants from multiple different companies. Before I start, let me also introduce what we work on. Essentially trained as a computational biologist and formerly as a clinician. We work in the area of genome informatics and the variety of things that we could actually go back and look at genomes, trying to look at Mendelian genetic disorders. It's a very large active program. Looking at pharmacogenomics and cheminformatics, we run one of the largest consortiums in this area. And of course, looking at genetic epidemiology in terms of looking at multiple genomes from across the world, South Asian genomes as well as the Middle Eastern genomes. And we are also one of the partners of the Pan-Asian Population Genomics Initiative, which looks at genome scale data across the South Asian population. And in all these networks, we work very closely with clinicians, trying to understand their pertinent needs in the clinics, and of course try to solve by, of course, providing evidence, and of course in many ways developing technologies which are cost effective and scalable. And of course in the wet lab part, or in the basic science part, we work on the non-coding RNA. That's not what I'm going to talk about. Most of what I'm going to talk about is essentially going to be in the area of pharmacogenomics, with some overlaps into the area of genetic epidemiology. I'll not need to reiterate this, but probably we were in the best times out here, because as you see, the cost of genome sequencing was dwindling very rapidly. The lab was founded in 2006, and as you see here, that was probably one of the most CISO experience and in terms of the exciting periods in genomics. But when we started our lab, the very pertinent question that we asked is equitable distribution of genomics present across the world? The answer is probably no. The top panel is essentially the genetic admixture plot from the human genome diversity. And what you see here at that point, this is way back in 2008, you had 6 genomes, or whole genomes at that point of time. This was the reference genome, the Bender and the Watson genome, and the Chinese, Japanese, and the Korean genomes. And what you see here is essentially all the genomes that were sequenced at that point of time were from very homogeneous populations, or less admixed populations. And the large population that you see here, which is the Middle East and Central and Southeast Asia, which is one of the most admixed populations out there, and probably encompassing a third of the whole human race, didn't have any genomic representation out there. And that's what we started way back in 2008-2009, sequencing the first human genome from here. It's today probably the 10th anniversary of the first Indian genome. But of course, we didn't really stop there. We went across collaborating with multiple different partners, including the Sri Lankan, the Malaysian, and today we have the largest databases for the Middle Eastern population, which is called Almina, which is published and available, and South Asian populations called SAGE. And very recently, we completed 1,000 Indian genomes in collaboration with Dr. Thangaraj out here at CCMB. Now, the real question is, of course, it's not a question that whether you can do genomes anymore, but how can you really put it back into clinical practice? And there starts a problem. And the problem is defined as here. In most cases, all of clinical medicine is based on average patients or average response of patients as defined by the clinical trial. And in most cases, a clinician is really clueless of what this individual is all about. And this is very contrary to what the fathers of modern medicine had originally stated. Hippocrates stated it's far more important to know what person the disease has than what disease the person has. But having said that, practically all of modern medicine that I learned in my medical school was to essentially look at the disease and treat the disease, rather than essentially look at the person and look at the nuances of what are the individual variations in different individuals. Now, there are two problems with treating diseases and not really individuals. The first problem is that not all drugs that you give to a patient is as efficacious as you would believe. And to give some numbers, approximately around 38% of individuals would not really respond to, would not rather respond to a class of drug, for example, antidepressants, and this could be as high as around 75%, and this is published literature way back from 2001. And that's one problem, that you might in good faith give a particular drug or a class of drug to an individual, but the individual really doesn't respond because of course, diversity. This could be genetic, environmental, social, and many other factors. And the second problem is essentially much more severe, that you actually create a problem to start with, and this is what we call as adverse drug reactions. Of course, adverse drug reactions could be of multiple different severity, from type A to type F as per the international classification. And what you see here is what is known as Stevens-Johnson syndrome, one of the most what do we call detrimental kinds of adverse drug reactions. Now, put in numbers to this, of course, there's a lot of genomic data coming up on how to actually provide individualized therapies, and there's an area what we call as pharmacogenomics. And the field typically grows today at around 4,000 to 4,500 publications every year, and the corpus of information available today is approximately around 20,000 publications. Now, that's very evident that no single lab can actually go back, or no one single individual can actually go back to all the evidence and sort of put it back into clinical practice, and that's when you need to have an approach to look at crowdsourcing. But of course, to understand these genetic variations, you need to first go back to physiology, and it's very simple. Almost all the drugs that you take is through the gastrointestinal tract. It undergoes metabolism in the liver, which is called the first pass metabolism. The drug is distributed in the systemic circulation. They're converted either from an active to inactive format or from an inactive to an active format. And of course, there are other two organs which essentially excrete. One is the kidneys, which excrete this metabolize in the urine in a soluble format. And of course, as I said, the liver. And in very small cases, you could actually inject the drug directly in the systemic circulation, and these drugs distribute in the extracellular space, and cells are in this extracellular space or in this fluid, and then of course they extravase it back into the cell and then into the cognate target sites. Now, to make this very simple, you have the drug, and the drug needs to reach the cognate site of interaction, which are essentially proteins. And before the drug can reach the protein interaction site, it has to cross multiple layers. And as you see here, multiple layers here from the gut to the liver, excretion, so on and so forth. And of course, it has to cross an array of metabolizing enzymes, because these are xenobiotics, and therefore body is built to metabolize them in inactive formats and excrete them. And after crossing these barriers is the molecule that is available in the extracellular space, which can now come into the cell or the cell surface or into the cell and actually have its cognate effect. So practically speaking, we are talking about proteins which are either carriers or transporters or metabolizing enzymes or the drug targets, and genetic variations in any one of these components which can affect the carrier function or the transporter function, the metabolizing enzyme function, or the drug target function could actually affect how the drug is metabolized, excreted, and acts upon a particular individual. Now, to give a very simple example, I'll give one example of a drug called clopidogrel. This is a prodrug, which is an antiplatelet activity. So it's taken as a tablet, so it's absorbed by the intestine. There's a set of enzymes or rather proteins which actually modulate this absorption. It's distributed in the systemic circulation, where it's metabolized and excreted into inactive format by extraces, and the excretion is modulated by the kidneys. And then it of course goes through the liver, the first pass metabolism. An array of cytochrome enzymes could make it into an active format, and the active format is essentially going to the platelet surface and bind to the P2RY12 receptors and modulates its antiplatelet activation. So as you see here, there are multiple different genes which modulate the absorption, the excretion, the metabolism, and the active conversion of this particular prodrug, and therefore genetic variations in all of them could actually modulate with the pharmacodynamics or the pharmacokinetics of this particular drug. Now, this is very well established in literature, and this is probably one of the most well studied what I call pathway models for pharmacogenetics out there. So we went back and looked at the population scale data, and this data was from Dr. Dwaipan Bharadwaj. You heard him yesterday, was our former colleague. And we looked at the Indian population and asked this very simple question. In many facts, the Indian population here is just 5 states in North India. And we asked this very simple question: are there allelic differences in each one of these genes that I described before, and are these allelic differences very distinctly different from the world populations? And the world populations are essentially from the 1000 genome and the HapMap populations. Now, what you see here is quite distinct difference in the North Indian population compared to the rest of the world, and what this would mean is that we have a better intestinal absorption, a better bioactivation, and a better inhibition of platelet aggregation. Or in other words, probably the drug dosage that we are going to give for an average individual out here in this allelic background is probably much higher than what is anticipated and trialed in this allelic background. And this is just one example, of course, where you can show that there are allelic differences in any one of these pathways which could affect not just the activity, in many ways the overactivity and adverse drug reactions to certain drugs. Now, of course, you can do this on a global scale. There are multiple population scale genome data which is available currently, and what you can essentially classify individuals as an ultra rapid, extensive, intermediate, or poor metabolizer. And as you see here, if you move from the left side to the right side of the screen, you would see this, and keep note of the red pies out here. And the red pies is essentially a poor metabolizer. And as you see here, if you move from this part to the rest of the world, you would see the red pies increasing on a gradual fashion. And what this would mean is also that a clinical trial probably done in this one particular population and the dosage determined in this particular population might have a different dosage requirement in this different population depending upon the allelic background and the metabolizer status of that particular population. And therefore, population genomics can actually provide very interesting insights into probably tuning or fine-tuning the drug and the dosage for a different population. So to do this, what we did is to essentially go and involve a large number of students, around 200 students, mostly undergraduate level, to collect information from literature in a very systematic format, and of course build softwares and tools to be able to ease this interpretation, because no human being individually can actually learn this corpus of information. And what we built was essentially a very complex map in here, and I'm sure nobody can understand this map, neither can I. Very simply put out here, these are all the drugs, these are all the drug targets. All the drugs needs to reach the drug target, and before doing that, as I stated before, they have to cross two barriers, one of metabolism and one of transport. So you can trace each of these lines out here and actually trace their way back to the drug target site. Now, this is nothing that any single individual can comprehend, because this is essentially the corpus of information, all information put together for all the drugs and therapeutics out there. And so what you do here is to essentially overlay genome scale information, and as a pilot what we did was to do for the Malaysian single genome, this was second or third genome that was sequenced, and essentially put this information in terms of the variants and look at the variation which could affect any one of these processes, either the transport, the metabolism, or the drug target site. And essentially what you could create is essentially a Google map for the entire population for this single individual. So you could classify all the drugs out here by the therapeutic class and all the drug targets out here, and you could trace each drug, which is each line out here, from the drug class to the drug target site. And if you have a colored line here, that means that you have a roadblock. So this is essentially from moving from point A to point B, and you can look at the roadblocks depending upon what is the genetic makeup of this particular individual. Right? So you can look at this subclass of oncology drugs who would have no effect with the transporters, no effect with the enzymes, and therefore absolutely no issues reaching the drug target site as expected. But of course you have a subclass of this oncology drugs which would have problem with the transporter, which would have problem with the metabolizing enzyme, and therefore probably suboptimal doses reaching the drug target site. So this is for one single individual, but of course to build this you need to do whole genomes for individuals, which is not really possible in today's world. So what you could do is to really look at population scale data of whole genomes or whole exomes and actually create this map for a population level for a cluster of individuals and ask very pertinent questions of what are the classes of drugs which require to be tested in that particular population, even if there was a testing that was supposed to be possible. We looked at two large populations, or distinct populations rather, not large. One is Malaysia, which is a quite homogeneous population, and of course a very small country in the Middle East, which is called Qatar, which has around 150,000 natives of Qatar and a population of around 1.3 million. And the idea was to really look at two distinct kinds of populations and ask this question, are there ethnic differences which could be promulgated, or are there principles that could be looked at looking at population scale genomes out there? Now this is for the Malaysian genome. This was around 100 individuals, and what we found is that a certain set of drugs for which there are certain set of variations which are distinctly different. So this is the Malay average allele frequencies, and these are the allele frequencies of the 1,000 genome populations. And you could see here is a very finite set of variations which had distinct differences with the world populations out there, and therefore this would be important to test only this finite set of variations which are distinctly different in terms of the allelic frequency and probably, and therefore you could build cost-effective test. In this particular case is just 55 variations. You could just screen for 55 variations or subsets depending upon what are the class of drugs that you need to give to individuals. And of course you could create maps for a population scale. In this particular case, this is at a 10% frequency cutoff. So if the population has more than 10% of individuals with any one of these roadblocks, you could plot them on this particular map. And one example that I can show is for methotrexate. In this particular case, the transporters could be inactive in approximately 10% of the individuals. the metabolizing enzymes could be deficient in at least 10% of the individuals, and therefore would have an effect on the particular drug metabolism and activity in this particular population. And as a matter of fact, if you go back to literature, there's actually at least half a dozen of publications on methotrexate pharmacogenomics in terms of clinical activities and dosage in the Malaysian population out there, reiterating the fact that you could look at population genomes to get insights, and these insights actually tally very well with all the publications that have been published previously on this particular matter. Okay. And moving on to the Middle Eastern population, Middle Eastern population has never been studied in the genomic landscape previously. So we put together one of the largest databases today, encompassing around 26 million genetic variations, 2,000 whole genomes and exomes. And I'm going to present as very small population scale data for this country called Qatar. And in Qatar, I said the population is around 150,000 individuals, but show a very strong what you call segregation, in largely into 5 or 7 classes, depending upon what is their ancestry. And this is largely because they have a consanguineous practice marriage for ages altogether, and therefore the population could be stratified genetically into very small groups depending upon what their ancestry was. So you could look at the pharmacogenomic map as before, and this is one example where you can look at, for example, this particular genetic variation was very frequent in all the subpopulations of Qatar compared to the global population. In this particular case, the global population was the exact all, around 60,000 exomes. And you could also have examples where there are some subpopulations which have low allele frequency compared to frequencies which have, or populations which have high allele frequency. You could have populations or subpopulations which exclusively have a particular genetic allele in high frequency compared to the rest of the subpopulations out there. In other words, even for very small populations of 150,000 individuals, depending upon the practice of marriage, which is essentially a social practice, you could have very strong segregation even for clinically actionable markers out there. Now this is important because if you know the ancestry of an individual and the social practice is still to continue the practice of marriage, you could look at the ancestry of the individual and you can actually derive very cost-effective tests. In this particular case, for example, if you want to give tacrolimus, you just need to do one genetic test, and that genetic test only for this particular subpopulation of individuals. Now extrapolating this to India, which has approximately around 4,000 odd endogamous populations, right? You could easily understand the impact of how genetic diversity in terms of social customs could actually impact clinically actionable variants. So to put together for the Middle Eastern population, in this particular case Qatar, an average individual would have approximately 3 actionable pharmacogenomic variations. We could of course rediscover from the data a lot of drugs which are presently being withdrawn from the market because of high rate of adverse drug reactions. Now, having said that, Middle East has always been very close to India, because a large number of populations today in India, encompassing close to around 10 million in total size, trace their ancestry from various regions of the Middle East, and therefore, and they still follow the practical custom of endogamous marriage practices, in many ways consanguineous marriage practices, and therefore many of these genetic variations which are enriched in here are also enriched in the subpopulations out there, and therefore looking at their ancestry and looking at the subpopulation for very specific clinical markers are therefore very important and pertinent. Going back to India, of course, we assimilated this large data set from multiple different studies across the country, from collaborators as well as from public data. And there are a few applications that you can derive in deriving cost-effective diagnostic. For example, this 5-fluorouracil. This is an anti-cancer drug which has a rate-limiting metabolizing enzymes, which is a DPYD. So you could look at the DPYD variations which are frequent in the population. There are just 15 genetic variations. You could go back to the clinic, do a very quick sort of a clinical study to look at individuals who develop adverse drug reactions to 5-fluorouracil and people who don't. And this is done in collaboration with the Ahmedabad Institute of Medical Sciences. And what we find is that there are just 4 genetic variations in the population which has any clinical effect. This could be built into a genetic test. This test was offered for 2 years and now licensed out to one of the largest diagnostic companies. So this is one way where instead of sequencing a particular gene, you can just look at 4 genetic variations and then of course offer the test to patients. These genetic variations put together encompass around 8% of all people out there, and that necessarily means that people who have cancer, around 8%, assuming that they are all predisposed to cancer, would have this adverse drug reaction. Very recently with CCMB, we put together one of the largest studies in terms of the genome sequencing, and this is a very recent program which is called Indigen. Dr. Tangraj has been a very good collaborator. And the idea was to essentially look at a pan-India collection of genomes and look at allele frequencies to develop cost-effective genetic markers. I'll just give one example and close. There's one case of a disease which is very pertinent in a particular group of individuals called the Chettias. It's called succinyl choline apnea. It's in a gene called the BCHE, or cerebrocholine. There are of course multiple different surnames that you can sort of look at, and the population frequency of this is approximately 1 in 24. 1 in 24 individuals in the Chettia community would have this genetic defect which predisposes them to a condition called succinyl choline apnea. And from population scale genomes, what we could look at is to look at all clinically actionable variations for BCHE gene. And what we discovered is just 3 variations, not just one, but 3 genetic variations which are very frequent in the South Asian populations and of course in the indigenous populations. And therefore, you could create a genetic marker test for just 3 genetic variations which can now be put in clinics. This genetic test is presently being offered as part of the GOMED program. Just 2 slides, and it's also now very important to not just do the genetic test but also be able to communicate this test to individuals. So we have a genetic card and a genetic app that is being piloted at the moment. I'll stop there and take questions, but before that let me acknowledge a lot of people which made this possible. Sridhar for most of the NGS and the lab work, a lot of students past and present who contributed to multiple different programs, and of course fantastic collaborators who are part of the Guardian Consortium, and of course a very recent consortium, the Optima Consortium for Pharmacogenomics, and of course consistent funding from CSIR which made this all possible. Thank you.